I'd like to welcome Mike, who's going to talk to us about developments in sequencing technologies. So first, Mike, I'd like to ask you about when you joined the Wellcome Trust Sanger Institute, what was sequencing a genome like back in the 1990s?
It was very laborious and took large teams of people. I was recruited to do molecular biology and solve the problems with the Mycobacterium tuberculosis genome. That organism causes tuberculosis. It is a major worldwide pathogen, kills many people, still does per annum. It's got a genome of about 4 million base pairs. The sequencing technology used at the time, Sanger sequencing, could only sequence about 800 base pairs in one go. And we sequenced that genome in a number of stages. We couldn't sequence the whole genome at once. First of all, people had to make large subclones of the genome in 40,000 base chunks in what's called cosmids. Those cosmids were propagated in E. coli. We had a team of people that looked at each of those cosmids and worked out how the sequences within them related to each other across the genome, what's called mapping. We then took each one of those individual cosmids, prepared DNA from it, a process which took 2 or 3 days. We then had to do molecular biology on those 40,000 base pair long bits of DNA to make a library to prepare the DNA for sequencing. We made a library by fragmenting those 40,000 base pair long bits of DNA even further by smashing them with sound energy, sonication, into fragments that were about 2 kb long. We then introduced those libraries into E. coli, grew them up, plated them on agar such that lots of individual colonies grew, and each of those individual colonies was a template for sequencing, and that was a clone of an individual 2,000 base pair molecule. That DNA from that colony was grown up, DNA was prepared, we then did a sequencing reaction on it. So that process was very, very slow. Sequencing the genome took 3 or 4 years, took many millions of pounds, and a team of about 40 people. So that whole process was very laborious and took many years and many millions of pounds.
So what were the technology developments that allowed us to sequence genomes faster and more cheaply?
In about 2005, 2006, next generation sequencing, or what's known as NGS, was born. And I guess the prime technology there was Illumina. Illumina sequences DNA in channels on a glass slide. At the time it had read lengths of about 35 base pairs. With time now that's grown to about 125 on one of these sequencing chips. This flow cell has got 8 lanes, so it can sequence 8 samples at once. But each of those samples can be multiplex, can be a pool of individual samples that are each uniquely tagged at the library stage. And now we can sequence 384 bacterial genomes in one of these at a cost of around £2 per genome. In order to produce complete genome data, one of the enabling advancements has been long-read sequencing technologies. I guess the first of those came out in 2010, Pacific Biosciences. Pacific Biosciences sequences in one of these, a smart cell, SMRT, single molecule real time. At the bottom of this chip there are something like 150,000 wells. Each of those 150,000 wells can house the sequencing of a single DNA polymerase and a single template of DNA. That template of DNA can be sequenced in real time at something like 2 base pairs per second, and the read length of this instrument is something like 10 to 15,000 bases. So that read length is able to span some of the larger repeats in the genome, such that for a cost of one of these, £100 or so, in a 4-hour sequencing run, you can get something like 500 megabase of sequence in 10 to 15 kb chunks, which is, when you take that data and assemble it, essentially the genome comes out in one contiguous piece. So you get a finished genome in 4 hours for £100 or so. And the next generation of sequencer along from that is the Oxford Nanopore MinION, which is coming along apace, has been developing over the past few years. And this is a USB link sequencer. For about £400, you can run a small number of bacterial genomes on this sequencing flow cell that fits into here, that links into your laptop. It could be taken anywhere. You could sequence, people have sequenced on the space station, people have sequenced up mountains and in Antarctica and in jungles and even in the lab, etc. So, you know, you can, it's very portable and it's very enabling. It enables you to do new things. With this, you can analyze the reads as they come off. You can match those reads to a number of reference genomes in the database to work out what it is you're sequencing. So perhaps you've got a person with an unknown infection, you could take their sample from them, get some DNA, sequence it on here, and within 15 to 20 minutes of initiating the sequence, you can work out what it is they've got essentially.